It is a great pleasure for me to introduce our BRAZ webinar guest speaker, Dr. Arnaud Becheler. Dr. Becheler is a postdoctoral fellow at the University of Michigan, mainly working on simulation models for population genetics and phylogeography, with renowned scientists as Professor Lacy Knowles and Professor Craig Moritz. He took up his bachelor degree in life sciences from the University of Bordeaux and received his PhD from the University of Paris-Sud. Currently, Arnaud is interested in modeling the relationship between environment, demography, and genetics, as he will explain to us. For this, he works in a multidisciplinary team, collaborating with biologists, mathematicians, statisticians, and also software developers. Aside from his talent as a bioinformatician, Arnaud is also an excellent drawing artist. And he reunites these skills in a blog that's called Derek and Leonard. I hope I'm saying it right. And in this blog, he communicates about programming concepts and culture. Please join me in welcoming Dr. Arnaud Becheler this afternoon.
Thank you so much, Cecilia, for this really kind presentation. Thank you, everybody, for being here, for having invited me. It's really an honor. I'm just very excited about this presentation. So I chose to present to you a kind of a wide conceptual map of the field I'm working in, because it's so easy to dive in details. I didn't want to lose people in the code or lose people in statistics, so I tried to summarize that. So if you feel like I'm staying too much above, ask me questions at the end to ask for precisions. It would be my pleasure to answer you. And the general question I want to answer here is when you're interested into simulating genetic diversity in heterogeneous landscapes, you have a set of questions that arise from that. And this is why do you want to do that? How you want to do that? And what are the limitations of these kind of approaches and questions? These limitations can be of different orders, technical, cultural, statistical, and I will just give some hints about that. And also at the end, I will try to open a discussion about what I think are the perspectives of this field, where I think we should go, where I think we are heading towards, and what we could do as a community of researchers. So first, why heterogeneous landscapes? So this is actually maybe the first scientific question I was asked by a researcher when I was doing my master. I wanted to do an internship, and I was really insistent about doing this internship with space, explicit space. And he asked me why. And at the time, I didn't really know how to answer that. I said, because that's the way I think about things. Now I came with a bit more explanations. So heterogeneity in landscapes have two dimensions. First, a spatial autocorrelation. That's the idea that when you look at areas in nature, the closer they are and the more similar values they tend to have. For example, elevations in point A tends to be more similar to point B when A and B are close. But there is also temporal autocorrelation. That is events that happen in quick successions tend to be more similar than events that happen when they are separated by a long temporal distance, a long time. For example, you can think of dynamics like climate changes and vegetation dynamics. And these two processes can have a profound effects on the different scales and levels of biodiversity. And that's the question that, for me, like, it's a very like subjective view, but I really like this question. I'm just enjoying exploring that. So an example about this question is how to explain the biodiversity of campos rupestres, that are landscapes that happen in that occur in southeast Brazil. And they are characterized by different features that you may know much better than I do, that are a higher elevation, above like 900 meters. You have poor rocky soils, and that forms a disjuncted mosaic of sky islands. And something interesting about that is that even if it's just 1% of the area of Brazil, it's still like 15% of the plant species number. I'm like, when I discovered these numbers, I was really like really amazed. It's impressive. And one factor that can explain that is that these areas seem to be climatically buffered. That is, in terms of climate, they seem to be more stable. And one question that we can ask is, can we infer, what can we infer about past processes? And for that, I will give you like a tiny animation about what I'm thinking about in terms of dynamic models. So that's a little model that I coded last week that simulates how a population could spread through this disjuncted mosaic of suitable areas. So what happens here, what you see happening is that there is a climatic pulse that relaxes the connectivity of the unsuitable areas of the matrix, and that allows like the species to colonize further suitable areas. And when the climate pulse ceases, like stops, the connectivity between the sky islands decreases, and that makes migration impossible again. And this process continues for quite some time, and we will see like several pulses happening. So these pulses are that have been like widely researched. That's a very well-researched field, but it's not that easy to actually know what exactly happened in terms of demography. Here you see a second pulse that happens. That is like, it's quite difficult to actually know the parameters of this simulation model that you see. I just like calibrated that to have a nice figure. But the question I want to ask is, if I really want to focus on a particular species, that could be a plant, like Cecilia studies orchids, some people study mammals. Like, if I focus on a species, how much can I inform these past processes based on some data I would have? So that is the data that we have. In my case, they are genetic data. And this question would be, can we infer, like, how much details can we infer based on that? So there is like a whole field about these two, inferring climatic oscillations from, like demographic oscillations from genetic data. So I put you some references. All the references are at the end of the presentation. I will make the presentation available if you're interested. And all the animations are also available online. Yeah. So why would you use genetic data for inferring that? So think about the past, like the past processes as being long gone. You don't have much historical data about past distributions, about past demographies. And if you have, like with fossils or whatever, you can find observational, like all the observational data, they are very scarce, like very sparse and biased at best. Like that's a problem with this kind of data. Although the present may be simply unreachable, when you look at this kind of processes, some ecological or evolutionary parameters or processes, you cannot measure them in a lab, even in a very controlled environment. You can measure things like mutation rates or fecundity, for example. You put an insect in a tiny box and you look at how many eggs they lay, and you see how this number, how much variation there is when you change the temperature, for example. So some parameters you could hope to estimate that in a lab. Sometimes you don't have the money, you don't have the time, you don't have the material. And for some processes, like for example, the dispersal of a mosquito, like that would be a tricky quantity to estimate in a controlled environment. They try to do that, like with mammals is easy, they are big enough, you put a chip and you track them. With big enough insects, like some people, what they did was like, for example, connecting a wasp to a tiny windmill and seeing like how far she could turn. That was bad for the wasp, but they could at least come with an estimation of the dispersal parameters. But for like parameters that relate ecological quantities to like broad scale demographic processes, it's not that easy to do. And that's the promise of demogenetics. That is to use genetic data because you hope that at least some signals of past demographic events may still be conserved in neutral areas of the genome. I insist on neutral because for that kind of problem, you don't really want to have like selective pressure to come and just get entangled with your problem. So you want to put like selection a bit away and you focus on neutral areas. And these signals tend to take the form of patterns and spatial distribution of genetic diversity. So the whole question can be reframed like a level of abstraction deeper by trying to know how to use such an indirect source of information to gain knowledge about distal processes like climatic pulses. And that's what the field tries to answer. And again, you just open the box with multiple questions inside. What data do you want to use? At what scales? How do you want to formalize these processes? That is like, what is the model you want to use? And if you have the model, how do you use that to actually extract information from the data? And that's the question of the inference method you want to use. And once you answer this question, you still have like the technical aspects of how do I concretely implement the method on my computer, on a cluster, what softwares, all of these questions happens. So I will try to give you some idea of them. What data and what scales. So when I began my PhD, it was one of the first question I had. That is like, I began to work on Vespa velutina nigritorax. That is an invasive species in Europe. And at the time, it was like a very recent process. The first encounter was in 2004. The expansion was going really fast, and it had like economical and ecological impacts, mostly on honeybees. And the amount of data we had was extremely limited. It was like a tiny spatial sample of 84 females. People had like tried their best to come with like some markers. And at the end, we had like, the beginning of my PhD, that's what we had. We had like on the left, some idea of the distribution of Vespa velutina in France. It was an ongoing process. And you have like some sampling points, latitude, longitude, and then like some allelic markers at multiple loci. I just put 2 loci here. But that's the data you have. And what you hope is like to come with tools and methods that are able to inform for like parameters like growth rate, dispersal rates, these kind of things based on both observational and genetic data. And you fast, because if you do that fast enough, maybe that adjacent territories that are not yet invaded could adjust their policies and their control methods. So that was a very short process, small spatial scale that is like centered on Europe, on France. It's like, it's not the whole continent. It's short time and few data. At the time, the tools to do that just did not exist. Today, if I had to do that again, I would do very differently. In 6 years, it's just impressive how the field just changed. But at the time, I came with my own tools and my own models. So the models that I began to think of were already existing in the literature, but not generic enough for me to apply them. So I tried to like generalize them a bit more. Generally speaking, the central concept of the kind of model we're talking about is the landscape. What kind of data you use to represent the landscape. These are environmental data, like geospatial data, that you can like collect online, that informs you on like topography or like environmental data like rainfall or these kind of things. They are basically rasters that are like geospatialized grids of values, and you can have a whole set of that. What people do is that they kind of contract, like summarize this number of layers, like when you have rainfall, temperature, mean temperature, seasonality, these kind of things. They summarize that by using what is called species distribution models that are correlative approaches that try to give you an idea of what are the suitable areas for your species at hand. They correlate environmental values with observational data. And that's how you can get a map that is called a suitability map. And people have used this kind of suitability maps to link this suitability that takes values between 0 and 1 to ecological processes like dispersal, for example, with the terms of friction. Dispersal will, for example, you can say that dispersal will be very difficult when you go towards higher elevations or like towards like very unsuitable areas. And you can say, for example, for niche functions, you can say that the growth rate of the population follows like a given function of the temperature or the suitability, or generally the suitability. So in this kind of model, two parameters that are important are the growth rate and the carrying capacity. These are like the main way to think about local population dynamics. And then you have, on this geospatial landscape, you have your genetic sample. You sample like your gene copies at certain locations, longitude and latitude. And what you're interested into is, conditionally to this demographic process, demographic quantities, you simulate, you want to represent a demographic history through this landscape. And then conditionally to this demography, build a coalescent tree. The coalescent tree represents, that's this one here, it represents the shared genealogy between sampled gene copies. And it's very useful because like conditionally to the tree, you can like put mutation models on the branches and just get an idea of genetic data and like the spatial distribution of genetic diversity, right? When you think about these generic models, again, there are some challenges. First, you need to better identify some models. Concretely, what dispersal function do you want to use? Here, like you can see, like it's a tiny function that gives the probability to move from one point to another, and that's a function of the distance between like the departure and the arrival. Here, I represented a function that is called a fat tail, that is like you have a high probability to disperse at a distance, but then the probability to go very far is never collapsing to 0. You still have some probability to go very, very far. So these are like long distance dispersal events. But you have like other ways to represent migration that could be like suitable for different species. You can think of a Gaussian kernel that is like very high probability to disperse very close, but then like almost null probability to go very far, like just the probability mass of the dispersal kernel like falls to zero very fast. So you have this question of what dispersal can I do? I want to choose for my model. Can I switch between these dispersal kernels? So these are the technical challenges of like how much variation the tools and softwares we have allow. So until recently, it was not great. Like you could choose into a small subset, but it's getting better. We will talk about that a bit further. And the second question is that even if you come with a more precise, more formal description of your model, you will have parameters that are not known with certainty, like some parameters, as I said, are like very difficult to estimate in a lab. That means that you don't know their value. So how to inform them? It's statistical inference, and we will talk about that now. So you need to choose an inference method. In this kind of problem, generally you have at least some knowledge or some intuition about parameters values. That is like, for example, without genetic data, without nothing, if I ask you, what do you think is the average fecundity of a bear per year? I would say 2. Like I'm not a biologist, but I know it's not like 1,000. I know it's not minus like it's not a negative number. So I can constrain the values of the parameter. That's called a prior. And the fact we can do that encourages us to use what is called Bayesian statistics. That is like a framework of statistics that allows you to input some prior knowledge. But also, you cannot do much mathematical work with the model because of spatial and temporal variations in the landscape. You just describing your model mathematically and trying to extract an estimator of the parameter from this model as a function of the data. You cannot do that. It's just like in terms of formula, it's not very sympathetic. It's not very cooperative. And that's called an intractable likelihood problem. So you want to use likelihood-free methods. That is, if you cannot describe mathematically the parameters of interest as a function of the observed data, you can at least come up with a way to simulate them. That is like using simulations to explore the parameter space. You may be similar, if you are not familiar with the approach, you may be familiar with like a related problem that is sensibility analysis. That is like when you have a simulation, you go like through a grid of parameters and you simulate the output, and you have these colored grids of how the output changes with the values of the parameter. That's a similar idea to that. So here I will begin like a tiny series of animations, because if people are not familiar with ABC, that can be a bit tricky to understand. So that will be like the main background we will use for the animation. And I will explain that, and we will add layers progressively. So what happens here? Can everybody like see the animations? I don't want to present in the void.
Yes, we can see it.
Thank you, Cecilia. So you have on the x-axis, you have your parameter of interest. It can be the growth rate, it can be the carrying capacity, it can be both. I wanted to simplify the problem by just choosing one parameter, that is one dimension. And then on the vertical axis, you have the amount of knowledge that is available on this parameter. In this toy example, let's say that we have very poor knowledge of the parameter. That is, we know it's somewhere between 0 and 100, for example, the mean fecundity of the bear, if we are very naive. But we don't know exactly where. So we put a flat distribution, a uniform distribution, with a very low density of probability. You see the mass of probability that is like almost 0.1, and that's your prior distribution. The ABC method will use your simulation, like your simulation tool, your simulator, to randomly in this prior distribution pick one parameter value and simulate some data. So at the end of the ABC, you will have a huge collection of, I sampled the parameter and it's coupled with one output value. And we will see what to do with that. So return to the presentation. Yeah. So once you have like this cloud of simulations, like you have like all these couples, parameter and simulated data, how do you filter them? So the very basic algorithm for ABC is an acceptation rejection algorithm. What happens is that once you sample the parameter in the prior distribution and that you have simulated the data, you give a weight to this couple, like parameter simulation, that will be a function of the distance between observed data and simulated data. So this weight, if you are very strict, it will be 0 and 1. That is like you come with an idea of a threshold and you say, my simulation is close enough to the observation, I accept it, the weight is 1, or I reject it because it's too far and the weight is 0. This idea of a distance is actually quite interesting, and we will see that just after. So that is like the idea of acceptation rejection. Here you are exploring the prior, and that's every parameter is a new line. In orange, you have all the simulations that have led to a rejection, because in this orange realm, domain of like this orange region of the prior, it's very difficult to apparently to simulate data that are close to the observation. But on this region, you have like something different that is happening, that is like apparently all the parameters sampled here tend to accept the simulation to generate data that are close enough to the observation. But what is exactly the, like, how is it working under the hood? So that is, how do you define this distance and this threshold? And that was a big thing until recently in ABC, and that's still problematic in some aspects. So if you're interested in genetic data, you are actually interested with data that are very heterogeneous. You have missing data, you can have like different types of markers, and the dimension is very high. You have all these sampling points and all these individuals that have been genotyped and multiple loci. And what happens is that distances and metrics, when you're working in a high dimension, are acting like really counterintuitively. If you're interested into that, there is this nice paper from Agarwal and co-author, and it's actually very accessible, and it's kind of fun to read, because like I would never have thought that high dimension distances would be so tricky. So what you do generally in demogenetics approach, you decrease the dimensionality of your genetic data by computing summary statistics. These summary statistics are most of the time almost estimators of simpler models. Like you can use like the FSC, you can use heterozygosy, you can use like any function of the data that will condense a very large data set to one number. And you do that like for some, like as many summary statistics as you have, and at the end you condense the genetic data into a simpler vector of indices. The choice of these summary statistics is, again, until recently, it was really tricky, because when you reduce the dimension, you are losing information, right? So if you lose information that could have been used to estimate a parameter, that is actually not fun. Like you're like just like shooting in your own foot. So statisticians and mathematicians came with ways to avoid that. And we will see that later, but now the best approach you can do is to use RSunStat by Excofear and co-author. You compute as many summary statistics as you want, and now you just like give that to the method. But it's quite recent development. But the question that happens still at the end is, I will show you just before that this idea of the distance, how it works. Yep. here it's the same graph, the same process. The only information I added is now you see for every parameter you sampled in the prior, you can see in gray here the distribution of the simulated data. And in purple here, you have the distribution of the observed data. So think of that as your almost your target. That is like you want to target that. And the gray distribution has to be accepted. The parameter value has to lead to a gray distribution that has at least some amount of overlap with the purple distribution. So that's what we see here. This one has been rejected, 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 rejected. This one rejected. This one is actually was a close fit. So you accepted the point here. So, and as I said, here it's like a very simple data. It was like a toy example that I coded this weekend. So it's easy to visualize that. But again, when you're working with genetic data, it may be a bit more tricky. So now it's like concretely, what do you gain in terms of knowledge by doing that? And that is like the problem of the estimating the posterior distribution. As the ABC algorithm progresses and that you explore more and more the parameter space, what you hope, that's like statistician would spend hours on that, but what you hope is that the density of accepted parameters will at least increase in a region. Like you want one region to have like a more dense population of accepted simulation. And you also hope that the more simulation you run, the more your posterior density will converge towards a target. That is like, if I run like 1 million or 1 billion of simulation, at the end, it doesn't change too much my estimation. So you need to check that. Also, what happens is that as the simulations go on, and that you simulate data and filter them, you begin to update your knowledge by switching probability masses from unlikely regions to more likely regions of your parameter space. And what happens is that you can come up with point estimates. That is like when you compute the mean or the median of your posterior distribution. And that I will show you here is exactly that. You begin your simulations. When you have enough points, you begin to make like this density estimation of your accepted parameters, and you begin to see the more points you have, the better the estimation of your posterior distribution is. And we barely have few points now. Like it's not like we don't even need like 30 or 30 millions of points. You have like less than 10 points, and you begin already to have like at least some usable knowledge about the parameter. When you're using complex simulation tools with genetic data, like the order of magnitude now is like to use like at least 10,000 simulations or like 1 million, like in this order of magnitude. And if we let that run long enough, you will see that the posterior distribution will not even move. So at the end, you take the median or you take the mean of that, and you have the point estimate of your parameter. That is your updated knowledge about what likely values are. You simulated all of that before, like you had like all this degree of confidence, like a very low degree of confidence across your all prior, and now all of that has been switched towards like more likely regions. So it's the idea of ABC. In a nutshell, you could like spend hours and years studying that. Statisticians do that, and they have a lot of fun, and they're like really interesting to watch. So here what I did, just like based on genetic data, to give you an idea, is I said that genetic data have a higher dimension than what we were playing with, right? But you can still do some PCA at least. And you can, at the end, like represent the cloud of your simulations on a 2D PCA. And that's what I did here with the simulator that I developed. And You plot that, you get your cloud. Now you begin to have some idea of the inference step with more realistic data, because like the observed data I have is actually like this little cross here. I'm quite not happy with this figure, because ideally you really want the cross to be in the middle of your simulation cloud. Like here you have all these simulations that will likely be rejected because they are too far from this region where the observation is. So it means that either my model is not good enough to actually mimic the observations, or I just misspecified the priors. In this case, I totally misspecified the priors intentionally. I wanted to have some idea of how my simulator was behaving. But yeah, you want, like if I want to perform an estimation, I will try to come with like a more selective criteria. So what simulation tools can you use when you're interested in this kind of problems? You have programs like Splash3 that has been like, it has been here like for a long time. One of the most popular, it's extremely user-friendly. It's coded, sorry, in C++, amazing resource. And you have like other types of simulator. One that is like, that is going fine is Slim, that is the forwarding time simulation. So we have simulation programs, but sometimes it is impossible to get the desired behavior just by using these programs and adjusting them with the configuration file, because some details are actually hard-coded. When you think about details of the way that individuals move in the landscape, or details about the dispersal types, or these kind of details, sometimes it makes sense for your species. It doesn't make sense for anyone else but you in the whole world. You need ideally a version of a simulator that implements these variants. And what it means for the whole community is, I think that one big need presently is like to try to refine the granularity of software resources we have. That is, and I will just begin to switch towards the perspectives. I think we should go there for several reasons. As I said, we need more simulation models, and we need them quickly. That is like when you have a new need, for example, because a biological invasion is happening, or because like you have like an endangered species, or like, and you want to know more. You don't want to spend 10 years developing your simulator. You want it like fast enough to scale with the emergency of your question. And also, there are too much variations in the ecological process to hope that we will have a one-fit-to-all tool. Splash is awesome. It fits so many scenarios, but not all of them. That is, we need to be able to increase the complexity of the simulation models we use without losing too much time. And that has been recently eased by the emergence of new domain-specific programming tools, libraries, that is like snippets of code that we can reuse and combine. All of that may sound very familiar for like bioinformaticians that have like literally decades of advance. But until recently in this framework, like it was like non-existent. So what happened is that two frameworks that I find interesting is TSKit that you have here, that is coded by a big community of people, and Quetzal, that is like coded by just me. But it's cool because Quetzal, what it does is allows you to have like these kind of funny animations, but also to simulate complex demographies and to be able to reuse what exists as code snippets, but also like implement new snippets and interface them with the framework. So it's a bit difficult because if you're on a non-programmer, it may be a bit too complicated, but it allows you to generate new simulators that can be used by non-programmers. So that's a big advancement. One major drawback of Quetzal right now is that I did my best, but right now, in terms of simulating genetics, I can only simulate independent markers, that is like independent trees along the genome. that is problematic because we begin to have like, that has been going for a long time, a big trend, but like we are increasing the density of the markers. Like we have more and more like sampling points along the genome. And when they are too close, they begin to be like physically so close that their dynamics, across phylogeographic scales are not at all uncorrelated. They begin to be like more and more correlated. And that's exactly what TSKit does. That is for like tree sequence toolkit. They came up with like new ways to represent trees and new ways to simulate correlated trees along the genome. So you begin to be able to simulate more complicated processes more realistically and more efficiently. The question like that will make me busy for quite some time is, I really want now to interface Quetzal with TSKit. I have very little idea of how to do now, but I will find a way, I hope. That's the resource for Quetzal, with a few ideas about the code. Every time you code something new, you have to make a number of choices that make sense for you, and other people may choose differently. So in my case, I ended up like choosing C++ because it's a good language when you want to compromise between like how much granularity you can have in your code. That is like you have some expectations in terms of design, and also like you need to run like millions of simulations. So you need performances. One aspect of the choice that was relevant at the time is that I had like a person, like a friend, Arnaud, who was like very good in C++. And I knew that she could help me. So what she did, and I owe her so much. So also what C++ allows to do is like to give so much information to the compiler that you can detect a lot of mistakes before your code even run. And I like this aspect. So consequently, I largely use like a concept of generic programming for like people who are familiar with that. I used a lot of templates and metaprogramming techniques. I wanted to focus on modularity and user experience. And I think I achieved like some reasonable results in terms of that. But again, it's a C++ library. So if you don't know C++, you may be a bit stuck. So that's why I'm presently coding simulations that are called Quetzal eggs, and that are like much easier programs to use. Another axis where I think we are going is that we need easier workflows. Having tools and methods is awesome, but it doesn't really solve everything. It is still very difficult to understand and gather and install and configure all of that, all the required resource. If you need like to ask your new PhD student or to ask your new intern to perform an ABC analysis, the time for him to install everything, like that will take a lot of time. So, and to understand that, it will also be like more difficult, longer. So what we need to do, I think, is to decrease and hide the complexity of simulation-based inference frameworks, at least for demo genetics. And I think it begins to be enabled by like the disparition of like some technical locks. So recently, like not that recently, but like again, they published that during a long time. Random forest ABC is a new way to make ABC, like that's a popular way to make ABC. What it does is that it bypasses difficult aspects of estimation of, that is everything that is about calibration. That was, remember, like what summary statistic you wanted to use, how much information you were losing. All of that now is like handled by their method that is like just phenomenal. Also, it alleviates the computational burden, so you can like decrease the number of simulation you need, and because you decrease the need of calibration, that means that you can come up hopefully with more standard statistical workflows that you don't need like users to very finely tune. A more technical aspect is Docker. For those who know, sorry, because I will butcher the explanation, but for those who don't know, see that as a way that we can package and run anywhere entire environments. So like you can run things on Ubuntu that have been like coded on Windows, and like it totally like relaxes like all these constraint of I cannot install your program. And I think it simplifies the distribution and the dependency management of scientific softwares. Like concurrently to that, there is Singularity. That is also a containerization article. Like if you want to know more about that, go to see like this presentation, this article, sorry. It is like, I think the most impactful, for me, the most meaningful effect is that it makes Docker containers available for high throughput computing. That is like, if you want to run things on using Docker on your local computer, it's nice, you can test the tools. Now, if you want to publish something, you need to run that on a computational grid, on a cluster. The problem with Docker is that you cannot really use Docker on computational grids, or at least like that presented like big security problems. And Singularity helped to resolve that problem. So now what I'm doing is that I'm using Open Science Grid, that is a very heterogeneous grid of computational nodes. And these nodes are like in Europe, in America, and like all across the planet. They run on a diversity of systems that is like, it's very heterogeneous, but it's surprisingly easy for me to go there and just like run my simulations, because all this heterogeneity is hidden behind like Singularity, Docker, and all the other tools. So I think that, and I hope that the direction we will be going for the field is that if we can use these resources to build entire reproducible workflows, that is like alleviating users from the burden of just installing and calibrating and just like struggling with scripts and dependencies and compatibility issues. That means I think as a community, we need to accept the fact that we will have many different profiles. You will have like very technical people who don't publish articles, but just write codes. These are considered in, at least in England, there is like this title that is research software engineer. And I think that's a tendency, a trend I would like to see grow further. And then between that and the actual biologists who just interpret the output, we will have, I hope, like the whole range of occupations and interests and talents. So I think it's mostly it. If you have like questions about precisions or like anything, just ask me. Thank you, Cecilia, so much for inviting me. Thank you for the NSF for funding me. Many thanks to the Open Science Grid. Thanks to them, it just made things like so much easier. And thank you to Lacey Knowles and her lab that are amazing colleagues. And thank you to all of us for being here today, meeting virtually. And keep targeting your best. I hope it was interesting.
Yes, it was super interesting. Can you hear me, Arnaud?
I can.
Can you see me?
Not yet. I will exit the presentation.
Let me see. Okay, here I am.
I can see you now.
Yeah. So now we are open to questions. Anyone would like to make a question to Arnaud?
I can see the chat now. So if you're shy like me, ask in the chat and I will answer my best.
I have questions. So maybe I will start, and if someone has some question, you can say it at any time. So Arnaud, you said to us that we could use all summary statistics. We can extract many summary statistics and give it to the software, right? Without picking specific summary statistics. But how to avoid overfitting? Would this, like taking many summary statistics, it would not give us the problem of having overfitting?
Wow, that's an interesting first question. So when you give summary statistics, I'm trying to reconnect the dots. When you give many summary statistics, it means that you're like increasing the amount of information that you're actually making available to inference. And it means that hopefully you are not losing too much information in the dimensional reduction step. After that, like packages and methods like ABC random forest, what they would do is come up with listing the summary statistics that actually matter the most by order of importance. And so some summary statistics will just like not be useful at all. And they detect that it doesn't really help in like learning the parameter. In terms of overfitting, I think I'm not a statistician, so I would really love to have a statistician right on my left. I think it's answered by the fact that in ABC, you can have some idea of how well your method performs by using cross-validations. The idea is that you simulated all these data sets. It's not like you have only your observed data, you have all the cloud of simulations. And what you can do is like pick one simulation, momentarily consider that as your observation, perform your estimation, and see if you correctly infer the parameter values. And then you put back the simulating point in the cloud of simulation and you pick another simulation. And when you do that many times, you can have some idea of how well you can estimate your parameters of interest, because you know the parameter you use for the simulation, right? You can have like a graph with true value of the parameter because I simulated that with this value, and then like estimated value of the parameter because I run my method on the simulation cloud. And if it's biased, you will see that happen. If you have no signal, you will see that happen too. So I think it's one aspect of the answer.
Okay, yeah, I think it is. As you said, if I understand it well, there is a step where you will make a list of your summary statistics and give it a weight, right?
Yeah,
so some of the summary statistics will have no value at all for
I can show you like some the form it takes. If I very momentarily I will share the screen. Desktop one up. You see that?
Yeah, I'm seeing it.
Okay, cool. So this is the kind of graph that ABC random forest outputs by default. That is like, here you have some stats, the PI index for like computed for a number of populations. You have actually many more summary statistics, but the rest of them were not even like important enough to be part of the graph.
Okay.
And then ABC like classes that by random order of importance. You have the posterior estimation too, and what the figure that I was mentioning is, let me find it back. Validation, where is it? Yep. Yeah, sorry, I cannot find it. But if you type cross-validation ABC on Google, you should see like these graphs of like points that are more or less aligned towards on a y equal x line. So yeah.
Eduardo has a question in the chat. Eduardo, do you want to open your camera and microphone?
Yeah, as I saw Eduardo question, he's asking, thank you so much. Like, I'm glad that you liked the presentation. Your question is, Quetzal and other templates, are they open for community use? It's an excellent question. So yes, it was the whole idea of Quetzal. So I will share again to show you the, where to find it, this top one. So here, if you go on my GitHub, I will talk mostly about Quetzal because that's what I do. But if you go on my GitHub, Quetzal Quality is open to everyone. So if you want to contribute, like send me an email, say hi, just pull a request or whatever. Like I'm very excited about this project. And you have here like the readme, the description. There are like some tutorials, some instructions, and yeah, and same for I wonder if by templates you meant the tiny simulators that I call the Quetzal X. These are like programs that can be run on your desktop, on your terminal, but on your computer, I mean. That is, you don't need to know C++. And here you have the instructions. I have like a list of available assumptions. Right now, I came mostly with 2 because they fit what I'm working on. My hope is that we can grow this list together. If people have like more idea of models or whatever, that would be fine. Nice. Other resources like Splash is available, you can download it, but the code is not available, so you cannot reuse the code. TS-Kit, you can see the TS-Kit framework. If you type TS-Kit framework Becheler, who is the main author, you can find the resource, and it's really nice.
And Arnaud, if someone is interested in to adapt your concepts, to do some programming with the simulations, would you be available for talking to them? Could them get in touch to you?
Oh yeah, please.
Yeah.
We can have a chat tomorrow if you want.
let's leave your email
super
in
accessible.
the chat. Yeah. Let's leave your email in the chat for people that might be interested. Juan Carlos has a question also. Juan Carlos, would you like to open your camera or we can read? Can you see the question from Juan Carlos in the chat? Thank you.
Okay. So
you can
Juan Carlos is asking, I can read the question if you want, or you can do it.
He says you can, yeah.
Okay. Yeah. I was wondering if those approaches would be suitable in populational genetic studies for endangered species that are separated by different ecotypes due to a homozygous genotype, parental inbreed, and so on. Yeah, that's an excellent question. When I was mentioning that if I had to do my PhD today rather than 6 years ago, I would do things differently. That's in this kind of question. That's the approaches I was describing were based on coalescence trees. That is, that there are like demographic assumptions underlying the reconstruction of the trees that sometimes are not suited to some questions. And when the spatial scale is small enough and the temporal scale is small enough for you to have like when it is small enough, like I think the best way to do things now would be like to use forward in time simulators like MS Prime, right? You have like forward in time simulations that are beginning to be available and efficient. Like when I began my PhD, they were not efficient enough. So it was taking too much time and it was not scaling well with ABC. But people coded things differently in the last years and they did gain a lot of performance. I would be really interested into knowing at what scale we should use one resource on another. It's not clear to me. Like you're talking about endangered species, how far back in time do you want to go? What processes do you want to understand? How many individuals you would have in your landscape? How much biological detail you want to know about them? I think that from what I understood about your question, like Splash or MS Prime would be more adapted for that. And there was a second.
So, yeah.
Ilker has another question.
From Ilker.
Who want to read the question?
I can read the question. So, okay. Ilker is saying that he liked your question or your talk. And he is proposing a quick question. He's saying that you showed a few simulations tools and probably you know some more. He was wondering if there is any specific tool you think would be more suitable to parameterize life history differences across populations. He knows some can implement different habitat preferences, for instance, but he was wondering what about more intrinsic ecological differences, such as mating system, age-specific reproductive output, sex ratio, etc.
Yeah, excellent question. I think in that case, you want to go towards not causal resources. I think you want to go towards TS kits and like their whole ecosystem, because yeah, I would go there. I don't know exactly the set of assumptions they are working on, but they have like just so many models there. I think you could find somewhere some things there, definitely. Mating systems, reproductive output, age-specific reproductive output, that is you want the fecundity to change with the age of the actual individual, right? I know that I didn't begin to work on that during my PhD, but I was working on like the kind of models that allow to do that. I know how to model that. I just don't know what kind of tools exist to simulate this. But yeah, if you need an explanation about like this, how such a simulation model would use, we can have a chat about that, and it's really nice. It's funny to draw on the whiteboard, but it's less funny to actually code it, I guess. Even less if you want to estimate the properties of these more complicated systems based on your genetic data. So yeah.
Arnaud, everyone in the planet is applying what they do for research to COVID. Are you tempted to do something about it?
That's a good question, actually. I know that I was like really interested by, like, when I was in France, we had like many presentations about biomathematics, and people are working a lot with these kind of models. There are some important differences with the models I'm currently using. And I'm trying to find a point of convergence. What I heard recently about COVID, for example, that makes me a bit worried. It was a very short information, so I don't know much details, but apparently there is a paper that just got out and that showed that you can not really hope to reconstruct ancestry, like the kind of genealogical information we're interested in. Like your likelihood to succeed this kind of reconstruction is actually quite low. So yeah, I don't know. I will see how this field evolves. I think we will learn so much with the COVID, like everybody working with that.
Yeah. And I heard that your group has some collaborations with Brazilians to study something in Amazon. Are you involved, or do you know about it?
I would love to. When I saw the little simulation of compost rupestres that I did, I'm like, wow, actually, it's so cool. Like,
yeah. I love it. And I saw that other people on the chat love it too.
Okay.
Yeah.
Cool. Yeah, I think that is like the questions that makes me feel more alive, I would say. That is like, okay, we have like these sky islands and like things like moved in the landscapes like long ago, things got connected, disconnected, like can we say what happened like that? That is just like so interesting to me. Like
yeah, I'm looking forward to implement this
I wonder
of code on my data.
Yeah, on what data exactly? I was about to say, I wonder on what species like we could like hope to say things, right? So yeah.
Sure.
Okay, go ahead, Miguel. Sorry.
No, I was thinking that many students in our program are just entering the master degree, but it was nice for them to take a look on how you can approach some questions with simulations, right? So even if they were not in this area of coalescence at all, I think it was inspiring for people that can get in this business with the approach of simulation, right? Was interesting.
I'm very hopeful in terms of like where we are heading to concerning simulations, because like non-simulation based like inference methods that are not based on simulations, like they are like powerful and easy and super important, but like they lack the flexibility sometimes we need. You can almost always come up with a simulator at some point. You can simulate something, and even if you're not a mathematician, like don't be scared. Go to talk to like statisticians and applied like mathematicians who are applying their knowledge to biological fields. My PI back in Paris was Camille Coron. She was a pure like probabilist, and she was totally like open and very able to like explain to me like mathematical concepts that I didn't know I would be able to know. So don't be shy, be curious.
Okay. Why did you choose the name for the software? Did you get to see the animal once, or the question?
So quetzals like used to be like my favorite animal when I was like, wow, like how can such a beautiful bird exist? And when I understood, so I have always been like kind of fond of mythologies and like legends and stuff. And when I realized that Quetzalcoatl matched the coalescence template library, I was like,
I know.
Let's go there.
Perfect.
Okay.
Arnaud, I have another question. as Miguel said, there are many students, people that are beginning their careers as bioinformaticians. And also we have people like me that already finished their PhDs, but are starting to use bioinformatic tools more often. So I would like to know, like, you are a more experienced bioinformatician. What would you say to us, like, that are learning bioinformatics? What do you think are like good practices for who is starting to program right now?
Wow. It depends on what. Wow, that's a very like rich and deep question. I could talk about hours with that. I have like hundreds of ideas. I would say if you're like just beginning with it, like the first steps of your programming, give a name like to every variable and functions. Don't use like ugly variable names. Like I have a friend, like she began to call like in her code, she has A1, A2, A3, A4. At the end, like 2 months later, she had no idea anymore what was who. So I think programming is like the step of like how much you conceptualize things in your programming has to be very clear. And when you have a variable that describes like a growth dynamics, call it growth dynamics, don't call it array 1 or v2. I think like doing that, you will begin to have like code that begins to be more literal and that will be much more attractive to the beginner. And so you can forget a little bit about I read the program and frustrating the programming activity is, because at least it looks beautiful and you can understand it. So now in my code, I can like read my code, and even if I haven't been there in a long time, it almost reads like English. And that's something I like. If you're like more experimented, I would say test and retest. Unit testing is a concept that programmers in the private sector have used a lot, and it begins to be incorporated into our academic frameworks, but it's the culture itself had to change to do that. Unit testing is basically like every time you design a function or like a tiny thing, you want to test it and you want to automate this test. So every time that you modify your code, you can relaunch the test and make sure that you didn't break your code by accident. And now there are so many wonderful tools to do that. Like every time you modify your code, use a version control. Like use Git, put your code on GitHub, document your modifications. Use Circle CI. So Docker and Circle CI are like frameworks that allows you every time that you push your modification on GitHub, you can automatically trigger entire like test frameworks that will go through your code and test, like run every test. So you can never really push a version of your code that is broken or not functional anymore. So yeah, testing is important too.
Right. Right.
Renato wants to speak. Renato, do you have microphone, camera? Go for it.
Yeah. Can everybody hear me?
Sure.
So thank you, Arnaud, for this excellent presentation. It was very didactic. It was very informative. It's not exactly a question. It's just a comment. I was thinking about exactly what Miguel just said. I always get excited when I see our work about simulation, and even either using simulations or developing simulators. And it's very important. We always get worried about which tool do we use to analyze some data sets. We have a lot of tools in bioinformatics, and we always get worried about, okay, which one is the best? And sometimes we forget that it's important to know how to simulate those data sets, to usually to really perform the parameter tuning. We can change some parameters and see how these parameters can change the outcome of our research. So I always get excited, and I just wanted to congratulate you for this work, and it was very, very good, very nice.
Thank you so much.
Where are you talking from, Renato? You are in Pará today?
No, I am in Belo Horizonte.
In Belo Horizonte.
Yeah.
Yeah.
But he's from the very north of Brazil.
Okay, wow. You're
Yeah, I am from Belém, Pará.
Yeah, we have recorded, and you have a slide where you presented some tools for simulation, right? And people has your email, so if they might need some help.
Yeah, I'm like, when you need help or like just like explanations, like just like send me an email, like ask for a chat. Like what you said about the importance of simulating data to understand them better. Like I began to go to this field just because I had a master class about exactly that. How do you simulate data? And the problem was very easy. It was like, we have an insect that laid a number of eggs. How can we compute the likelihood function? How can we model the problem? And how can you run inference, like make inference? And so that's how I was introduced to simulations and Markov, like that. These are called Markov chain models that are family of models that are like quite easy. And it's such an interesting and motivating entrance to this world. So yeah, begin small, begin with a small model and compute the likelihood, and it's very, very accessible for everyone, including non-mathematicians.
Cool. Cecilia, if they don't have more questions, it was really fun to have you with us, Arnaud. And it was a great afternoon here in Brazil. And it was great, a great event.
Yeah.
Thank you so much for having invited me.
Yeah, thank you so much for accepting it, Arnaud. And thank you, Miguel, for accepting my ask, because I am from other department. I'm not from bioinformatics, despite I use a lot the cluster. Exactly doing simulations. So I do a lot of simulations on the cluster. This is why I get in touch with Miguel so often.
Yeah, so people today got to know Fiorini.
Yeah,
you are famous. You are famous in the cluster.
Yeah, but that's it. I'm doing simulations, other kind of simulations, not the kind that Arnaud showed us today. But that's it. So thank you a lot, Miguel, and thank you a lot, Arnaud, and thank you all for coming.
Sure. Okay, guys. Thanks a lot.
Okay. So bye-bye.